So I'm Ima Suirats. I'm one of the chairs of the interest group on agricultural data at the Research Data Alliance. And I'm very glad today to be moderating this webinar with the title COPO, Extending the Frontiers of Fair Data in Agriculture. The webinar is going to be introduced by Etuk, Tony, who is a research software engineer at the Earlham Institute since February 2015, and he has been working on the Collaborative Open Plant Omics COPO project, which is a platform for plant scientists to describe, deposit, and retrieve data easily. This session was already presented during the IGAD event related to IGAD in Finland, in Helsinki last year, and is part of the IGAD webinar series that we are organizing now to make sure that even though, or even the people that couldn't join us in Finland can make sure, we can make sure that they can get information about what was presented, and that you can also interact with those that are working on this particular project. So said that, thank you so much, Tony, and also Felix, which is who is working as well in the same project, to be here today and ready to listen to your exciting project. Thank you.
Right. Thank you very much, Iman, and thanks everyone for taking the time to join this webinar today. Like Iman's rightly mentioned, I'm here with my colleague, my right-hand man. So if you just put something on top of my name and then you superimpose Felix Shaw. So he's been working on this project from the inception. Actually, he started this project before I joined sometime in early 2015. And Felix is an expert in computer science with a specific interest in machine learning, image processing, and all that. My background too is in computer science. So together, Felix and I and Rob, who is the project PI, we've been trying to, over the past 4 or 5 years, trying to bring some of these techniques now to develop a platform that promotes fair data. Today we'll be talking about agricultural data, but COPO is much more than that, as you will see in a moment. So the nature of this presentation and the whole session, how we will be looking at it, we will start with the presentation. So for those that were not at Finland, you've not missed anything. We'll go through the very short presentation that we had there, but it's going to be an extended form because after the slides, we'll take you to the COPO platform itself to show you how we're actually implementing this, bringing this fairness into agricultural data and some other data types that we're supporting in COPO. So we'll show you the wizard system that we've developed there and the file metadata, how we can attribute metadata to files or research object as the case may be, and then we'll open the floor for discussion and then questions. I'll leave the floor now to Felix, who will run us through the slides.
Okay, hello everyone, good morning, thank you for joining. As Tony says, I'm a developer on the COPO project. Me and Tony have now been working together on this for nearly 5 years. So as he said, my background is as a computer scientist with a special interest in machine learning and image processing, signal processing. So coming into metadata was something new for me, but so here we go. So COPO stands for Collaborative Open Plant Omics, and it was originally conceived as a way of getting data into repositories. So as I'm sure you all know, nowadays there's a very big push in science to make data fair, so findable, accessible, interoperable, and reusable. That requires certain things to happen, and lots of people talk about these things, but I think to date there aren't that many ways to actually implement these things. Okay, so by findable, obviously we mean that once a piece of data is in the public domain, it must be you must be able to get hold of it somehow using some sort of search term. Accessible, basically it needs to be public. Interoperable, meaning that somebody else can take your data and reuse it, or a computer tool or program can take that data and reuse it. And then reusable is, well, reusable. So it was conceived by Rob Davey, who is the PI on this project, as lots and lots of people were coming to him all the time with omics data that they required to be put onto the European Nucleotide Archive. And he basically thought, this can be automated or semi-automated, and once these kind of annotation of the data, which is what people were struggling with, was the annotation of this data. Once that bit has been computerized, there's all sorts of kind of fair implications for that, and different networks of knowledge that can be created between these different types of objects. So COPO does this by it's essentially a website. You log into the website using an ORCID ID, from which point you will create a profile into which you can upload data objects. So it might be omics data, so genomic, metagenomic, transcriptomic, that kind of stuff. It might be other types of object which represent the kind of broader context of your work. So if you're a crop researcher, you might have field trial data, things like that in spreadsheets. So, or you might have, if you're into phenotyping, you might have photographs of plants growing, these kind of things. You might also have presentations that you've done. You might have posters that you've given. You'll probably have some manuscripts. All these kind of things can be uploaded into the COPO profile, which represents the body of work essentially for a particular project. And from that point, you can then go through wizards, as we call them, to basically assign appropriate metadata to these objects. And that metadata is powered by community standards. So if it's something like MyAPI for the minimum information about plant phenotyping experiments, or if it's doubling core, or if it's CG core, or one of these different schemas that's an agreed standard, our wizards implement these schemas to make the metadata associated with the objects findable, accessible, interoperable, and reproducible. Okay. So originally it was just for omics data. That was the first instance, but now we've widened its applicability to lots of different types of data, which I mentioned earlier, and also lots of different repository types. So here's an example of some of the things that we support. So we have CG Core 2.0, which is the latest version of the schema used by the CG centres for agriculture and crop studies. Doubling core, which is a very widely used kind of generic schema for describing data objects. MyAPI, which, as I mentioned earlier, is for plant phenotypes. And then we have for kind of, well, obviously for the omics data, we have the ENA schemas. And then for more generic things, such as perhaps images or presentations or posters, this kind of stuff, we use the Figshare schema. Okay. So the idea is that COPO provides a sort of common unifying layer for the deposition and annotation of these types of objects. So no matter what type of data you're dealing with, the wizards and the UI and UX that you're presented with will be familiar. The controls are all kind of intuitive, and deposition to a number of different repositories is essentially seamless. Okay, so Here we can see the figshare, ENA, and then for certain types of studies, a lot of institutes are now using these kind of off-the-shelf repositories, DSpace, CCAN, and Dataverse, and we support all of those. So one of the major things that helps with interoperability and findability is the use of ontology terms. So an ontology is essentially a network of ideas that link together. So you have, it's basically a network of nodes and edges, and each node represents something, some idea. So you might have a node that represents a cat, and the node is essentially just a URI, a digital identifier, which encapsulates the idea of a cat. And there may be an edge between cat and another node, which encapsulates the idea of a mammal. And underneath cat, there'll be another node that encapsulates the idea of a tiger, for instance. And so by traversing up this network, you can infer that the tiger is a mammal just from the way that these nodes are arranged in this graph. And by applying nodes like this to data, we can start to unambiguously label concepts or essentially rows in a spreadsheet you can think of. So you might have a spreadsheet which is for crop trials, and a column is labeled HT or H or something like that. And the person who created the spreadsheet knows that that column represents height, but nobody else knows that. You might be able to infer it by looking at it, but a computer certainly can't infer what that means by just that label. So what COPO allows us to do is to actually take an ontology term for the concept of height from the unit ontology, for instance, and apply it to that column heading, which then allows us to unambiguously identify that column of data as being a plant's height. So this then makes the data in that spreadsheet findable, because you could search for that particular ontology term for height, and it would be returned saying, this particular spreadsheet has that label. And also makes it interoperable, because we can say, right, this spreadsheet of data about Chinese spring wheat and that spreadsheet about Chinese spring wheat, but in different parts of the world, are both labeled with the same ontology term. Therefore, we know that these two columns of data represent the same thing and can be taken as one. Okay, so these ontology terms are both, you can add them both to the file level metadata, so metadata that describes the file as a whole, and more recently we're adding it to kind of column level metadata, or even text metadata in PDFs. And this is all powered by a service run at the European Bioinformatics Institute in Cambridge in the UK called the Ontology Lookup service. So I think that's more or less all I have to say. So I think Tony is now going to, I should probably just say, if you want to use COPO, the live instance is https copo-project.org. That links to the live instances of repositories. So if you're actually depositing data, use that one, but be aware that it will go into a live instance. If you want to just play around with COPO, you go to demo.copo-project.org, and that is connected to demo versions of the repositories. So you can upload dummy data into those, and nobody's going to mind. We've currently got a paper preprint at that DOI in bioRxiv, and our GitHub repository is at that URL. I think the development branch is the most up to date. So if you go to the master branch and don't see any activity, it's not because we're not doing anything, it's because we use a different branch. Okay, so Tony's now going to go through and show you some hands-on stuff of what COPO actually looks like.
Right. Thank you, Felix, for that intro. And that's what we presented at Helsinki during the RDA plenary session. So, and what we have here is the demo instance, like Felix has mentioned, of COPO. So you can use this as your trial ground, as your playground now, just to have a feel of how the project works and whether it meets your needs and all that. So I'm just going to run us through different aspects of the project. That's the homepage of the project, and I could also open that's the live instance. So the demo instance kind of mirrors exactly what is on the live instance. So we normally point people here to play with it before going to the live instance. You could see some of the activities and where we've taken COPO to. This might not be up to date, but we talk about COPO a lot, and some of the collaborators now, people from Oxford and University in the UK, and from different other places like University of York and Embley EBI. Felix has mentioned that already. And yes. So to access COPO, like Felix mentioned, you need an ORCID ID. So if you don't have an ORCID ID, you might have to go to there. I've already logged into the system. That's why it's allowed me to see if I can log out. So if you sign in, so that would be the sign-in page. So if you don't have an ORCID ID, it should take you to ORCID platform for you to register a new account and then be redirected here. So that is what it does. Takes me to ORCID. I enter my ORCID credential, and then that redirects me back to COPO. And this would be the landing page of the application itself. We have this concept of work profiles, which are kind of a logical grouping of your bundle of work, your research objects. As you can see here, some of these profiles, or most of them, are very much omics-based type of data. So they do have the concept of samples, and you have the concept of data files. This might appear in most of the work profiles that you tend to create. The submission, that is where ultimately the goal that you want to achieve, because you want your data to leave your local system and go into some external or remote repository. And you can start tying in other research objects like your publication. Like we have a preprint now, we could enter the DOI there, or you can resolve to any digital identifier. And then you have the people here, which is collaborators. I'll just start by creating a fresh profile and then walk us through different aspects of the system. So the two things that you need to create a new profile would just be the title of the profile. I'll keep this to a test. Give it some context in terms of description. Not much of a context with test, but that would do. So that new profile has been created, and with the default template now. And this list, or actually this collection of research objects, we keep adding to it. By the time we refresh our system, maybe new items might pop up depending on the need of the community. But for now, I'm just going to go through data files, because I think the focus of this webinar would be on the CG core schema agricultural data type, which we'll want to follow that route of discussion through. So that takes me to data files here, divided into two main sections. At the left-hand side, I can choose to, my left-hand side now, I can choose to upload data file. And after uploading data file, I can now create a bundle. So imagine you having like 10 files to submit to a remote repository, the same repository here. So you don't have to start annotating them or tagging them with metadata one after the other. In most cases, what we tend to see that is that people come with similar metadata for SQL or a number of files that they want to describe. So you can do everything, bundle them up together and submit them as a single unit. And if you don't know what you're supposed to do, this is how the data files to the right pane. This is how the data file workflow would be. So you upload your data file, and then you create a bundle to group one or more related data files that you want to submit as a single unit. And you add data files to that bundle. And then finally, you add metadata to the data files in your description bundle. So I'm going to see if I can find some something to upload here from a desktop. Test data. I could just pick these two spreadsheets. And they're uploaded. You could see that. So at this point, I'm going to go to the next point in my workflow, which is to create a bundle. I'll call that test RD webinar. And that's saved. And I could select these two data files and drag them and drop in that bundle. Now you can have as many bundles as you choose to, depending on how your workflow is or depending on the logical grouping of your description or your data files for submission. So here you could access some of the actions available to you within this bundle. You could add, of course, there's another way of adding data files. So if I had some unassigned data file to the left-hand side here, I could also add them here instead of drag and drop. Another thing is you could show the data files here. We are already on that view. The important bit is you can now add and edit metadata, which is the very first step into fulfilling the fairness of this data that you're trying to play with. So I'm going to follow that through, and as you can see, we support a number of repositories, and repositories would also lead to the kind of metadata or standard expected by that repository. So these would be the metadata standard that a different submission pathway would be expecting. So we have the CG core, which I'm going to follow through, and as Felix showed us in the presentation, the different types of repositories and data standards are being captured here. If you don't know what any of this will stand for, you just hover over that information icon. It will tell you what that particular repository or metadata standard is all about. I select that. This is the wizard itself, which I've initiated. So it takes you through a sequence of steps, asks you questions, and depending on the user's answer, might take you down different pathways. So click the next. It's going to ask me for the type of information. So you could see at the right side here of the wizard, information product type. And it's also giving me some kind of housekeeping information, how the information I'm entering at this stage of the wizard is going to be dealt with. So these are the different types, information product types supported by the CG Core 2.0 schema. I could pick any of this. Maybe I could select one as a data file, and I continue. They also have this concept of subtypes. So a way of refining into a much more fine grain where the type of information product that you're trying to describe. So I pick spreadsheet because that's what they are. And given those two information I've supplied, it now presents me the form to start asking me tailored questions. So I'm going to, this is just for want of a good title. I could copy that, and for description too, and paste that there. So you could see the constraints that are being imposed on these fields. If I go back one stage, You could also see that some of this could be recommended, some of this could be the fields or the questions that are being asked. It could be optional. So you are being informed on the constraints or the restriction that we put on the different fields. The language, I will stick to English, language of the day here. License. So like I've mentioned, at any point in time, if you don't know what each of these fields are asking for, there is this tooltips here to tell you exactly what is being expected. I'll just put in something there. So now it asks me for the creator. I could see what that actually means. Please give the name and check the correct spelling of the creator of this information product, I suspect. So I will stick to Tony. I will use, again, creator affiliation, and this is control vocabulary of all the different institutes in the CG group. These are the CG centers. I'll pick Hickory site, and I save that. Contact point, again, tells you what it's expecting. I could create one. Place the contact point for this, okay? Contact email. I'll pick one there. And I save, so that's added for me. I can progress with the stages. Creation date. I can populate that for the day in the past. Very futuristic I am. Embargo date. I could put some date that after this date it will be released to the public. Has metadata for, this is a way of linking to other objects that you may have submitted. I think so, to some remote repository. So I'll just put some identifier there. So at the end of the wizard, you do have this kind of tabular representation of your information. So you would see that going through the stages of the wizard, although I'd selected 2 data files in my bundle to describe, but it was showing me forms, kind of building up a template for me to put in metadata that will apply to these different data files that I have in my bundle. But at this last stage of the wizard, I now have the opportunity to edit information for the specific data files now, for the individual data files. So to do this, for instance, I could change the language here to maybe French. So this is just a way now to kind of select specific metadata for the different data files that are in your description bundle. And by the time I'm happy with everything that I have here, I could proceed. And it's just telling me it's the end of the wizard, and I could choose to submit this. So here you have the 2 data files that I want to submit. It's left there. The data file module, it's now taken me to the submission module. So here I could choose specifically the repository, the endpoint I want this metadata and the data files to go to. So you have the CG code, the different repositories that we've added to the system, and you select one of this repository and then you hit the submit button, and then it should allow you to submit this. So that is how the workflow, and by the time you submit that, it comes back with an accession, which would be in the form of, I'll show you exactly how that is for some submissions that I have here. So these are completed submission within a different context now. I think this one was for omics data, but this is how the accessions or the public identifier or the persistent identifier coming back from this repository would look like. So you will have these accessions here, and you will have the confidence that if you picked one of these sessions, if they're public, if you've made them public, you've lifted the embargo and searched them online on that repository, you'll be able to find them, and you should be able to cite this on any manuscript that you're sending outside. So that's one submission pathway, and you could follow a number of submissions pathways. And if I wanted to submit omics data, I could have followed a different submission pathway, which I'm just going to briefly touch on without dwelling specifically on either of those. So for the omics one, you could see here, they have the concept of the samples, which again, you could have your wizard to enter samples which you tend to sequence and then get the raw data file, and then you submit the raw data file. So I'm not going to follow that through. I'm going to discard that. But what I just want to hint on, which Felix would kind of expand more on that in the next session that we're going to run for the file metadata, is that Felix did mention about one way that we're trying to kind of advance this frontier of fair data in agricultural data is by marking everything with ontologies now. So you didn't see much of that when I was doing things around the CG core, but it's actually there in COPO. So for instance, if I wanted to describe this sample and give it an attribute like, what should I give an attribute of a sample like, if it's a plant?
NPK.
NPK. So I could search for that, NPK fertilizer. So that's resolving, as to mind back to what Felix mentioned, to the Ontology Lookup service provided by the European Nucleotide Archive, ENA or EBI. So that's resolving to that public resource now. So this is an ontology. This would be submitted along with your, this is part of your metadata. So you know without any ambiguity what you are referring to, because there could be a lot of ambiguity when people start to describe things. So you put in some value here. Again, this is an ontology field.
No, do 1 or 10, 10, then add 10.
10 again?
No.
Okay. So that, again, that resolves to an ontology. So if I were to click on any of this ontology, you'll see that it takes me straight, it resolves straight to where that thing sits in the graph of thing. And I use the concept thing. So it takes me exactly there, so I know what I'm referring to when I'm talking about. Okay, that's the value, and I could
like kilogram per head.
Okay, so I could. A what?
Hectare.
Okay, good. So again, I submitted the. So if you could see here the value, and then I'm giving it the unit. Again, all these are ontology fields resolving back to an ontology. So you could go along and describe your data like this. And one benefit of this is that, as well as being visible to humans, but more importantly, machines could also start to gather this information in a non-ambiguous way and start building a whole lot of knowledge product out of all these descriptions that you've done. So I'll pause here and hand over control to Felix, who would show us a different aspect of our metadata workflow within COPO.
Okay, so I'm just going to have to log out and log in as me. Do I need to clear the cookie?
No, I think it should let you. Oh, maybe you might need to.
I'm going to clear the cache. It thinks I'm Tony. How do I do that? They do cookies as well.
Okay. go back and still empty the caches as well. Good. Okay, so let's go back to full screen.
Okay, so I'm just going to sign in as me. We just had to clear the cookies because Tony's login persisted there. So what's my name? Not sure. That's it. Okay. Okay, so this is what my front page looks like. So demos, as you might expect. So I'm going to show you how you can start to annotate column level metadata. So I'll go into my data files that I've already uploaded, for instance. And we can see here we've got a load of spreadsheets. So what I can do is I can, what is this?
Somebody trying to.
Sorry. Right. So I can go into this view here in data files and click on this annotate data file icon, and we should see that the data within that spreadsheet is displayed in a tabular view like this. So I can now start to do some ontology lookups. So I might say, okay, so I want to label this column here. So I'd say, right, it's a genetic marker. And so that's gone away to the ontology lookup service, and it's searched, and it's found these two terms that could be applied to that. I'm going to choose this one. So I click on that, drag it to the column, let go, and now that column of data has been unambiguously identified as genetic marker information. Same thing with, I don't know if there'll be, I'll have a look for synonymous name. I don't know if there'll be anything for it. No, there's nothing for that. So maybe something like synonym. Synonym. That might be close enough. So we choose that. I know for a fact that there's a forward primer. Forward primer, single-stranded, oligo used for polymerase chain reaction. That's that. And we'll do the same for reverse primer. In fact, what I'm going to do, because I know I've used reverse primer before, I know it's going to be here somewhere. So reverse primer. So I've used that one before, so I'll take that. And this is something that I hadn't anticipated, the fact that when this list gets really long, you can't actually drag up. Okay, I'm going to have to search for it. But anyway, I will look into this, but also terms that you've used in the past appear here with the number of times they've been used as well. So if I do some overflowing with this column, we'll be able to just scroll through that column as well. Okay, but for the time being, I'll just do reverse primer. Primer. There we go. That one. Temperature here. Nothing coming for that. So you kind of have to work your way around this a little bit and just try and find the most appropriate term to describe what you're trying to do. And sometimes there won't, there just won't be a term. So this is looking up to the Ontology Lookup service, which indexes something in the region of 400 ontologies and something in the region of 4 million different terms. But, some things still aren't in ontologies. So we'll choose that. Stop that and then restriction enzyme. Okay, so we've got that. Okay, so we can see that we can quite quickly build up, you know, an unambiguous labeling system for what these different columns of data actually mean. Obviously, you can delete stuff and change things around. So, so this is now labeled. So we could go back to the here, and we could. Which one was it? 3? It was 3. Yeah, it was 3. Okay. So I could go in here, and then initiate a submission like this. Okay. Yeah, and then I could, as Tony demonstrated earlier, go through. Sorry, this. Yeah.
Clone that.
Okay. The reason it's giving me that issue is because that's a bundle that's already been submitted. Show data files, and then drag that. Okay, there we go. And I can say I want to add metadata, and then we just go through the wizard as we did before. And although it's not quite implemented yet, but in the coming weeks, that metadata, that column level metadata that I added, will be submitted alongside the file level metadata, so that people can then start to actually search for the individual terms within a file. Okay. I'll show you one more thing. I've got appropriate file for it. So have a look at this. This might just be being a bit slow, I'm not sure.
Yeah, so it's converting the PDF, right?
Yeah, this should be converting the PDF in the background into text.
Yeah.
Although it's not normally this slow, I wonder if there's some issue. Yeah. One more time. You know what? Could be. You've got any PDFs I could just test.
See if we can upload. Go to desktop. See. Digital innovation. There's no PDF.
Let's just go to. Sorry, I'm just getting a PDF because I'm just wondering if those PDFs that I tried to upload don't exist. So if I can just download a PDF from here.
Doesn't look like it. Okay.
There we go, download PDF. You're going to let me have it? No. Denied. Let's go to I'll try here. Let's have a look at Omics. And let's download this one. Download PDF. This demonstration may end in disappointment, I'm afraid.
Could it be our internet connection though?
Maybe our internet connection is being a bit funny. I don't know. In which case no one can hear us. Can anyone actually hear us? Yeah. Okay. So
Yes, yes, we can hear you, Tony and Felix.
Yes.
But,
Okay, good.
I'm just wondering if our internet connections.
Wait.
It's not that good.
Okay, well, what I wanted to show you was that. Oh, here we go. Right.
I'll just place it on the desktop, right?
Okay, right. Okay, so if I go back in to here, and hopefully we'll be able to upload it, although I'm not 100% sure. So if I upload that file that we just downloaded.
Attachment 2. Okay, that's it then.
Okay, choose that. Goes, and let's see if this is going to work. There we go. Okay, so this is now a view of that PDF, and we can come in here and we can start to actually label the text in this PDF. So let's have a look. So we have a look here. We can highlight the word rice and say rice. This goes off and it's found some different definitions of rice. So I can choose perhaps that one from food ontology, save that. And now we've got the label for rice in the project. Let's have a look, see what else we can find. Okay, so we found that they used ethanol. So we have a look for ethanol. I assume in here that, okay, maybe something like that. I'm not sure which process they've used, but there'll be something for the process they've used involving ethanol. If I knew a bit more about chemistry, I'd be able to choose the right thing. I'm just going to assume it's this one, and we can save that. Okay, so now we can see that we have these two things labeled, ethanol and and so you can go through and you can label up these PDFs basically as much as you like and as much as you think will make your particular research as findable as possible. So anyway, sorry, that was a bit of a convoluted demonstration because of the technical hitches, but I hope you've got the basic idea of what you could do to label your data within files with ontology labels. Okay, so I'm going to pass back to Tony, who's now going to, well, we're just going to basically open this to the floor and take questions and points that people have.
Yes, thank you so much, Tony and Felix. We have 5 minutes to go. We can simply open the mics to people if they want to speak, but since we have only 5 minutes, I don't know whether it's easier if they write the comments and the questions directly in the chat box. I see that there is already one from Simon Pierre. Tony and Felix. He's asking whether you could send some additional materials related to the app.
Yeah, so the best thing to do would be to go to the DOI. You want to bring up that DOI?
Okay, yes. Let's bring that up on the presentation.
Yeah, this DOI here is our preprint for a paper that we're writing. So if you have a look there.
Okay, yeah. Just testing that to see that's still the right way to go. So yeah, so that's the paper. So you could gather more information about the project from there. In addition to logging into the demo site and also reading the documentation, this might be a bit dated because a lot of things keep changing, evolving in COPO, but and sometimes the documentation don't quite catch up, but eventually they will. But yeah, those are the sort of resources that you have at your disposal to learn about COPO. Yeah.
There is also a question from Susie Robinson that actually I had this question as well for you, is whether it's possible to add additional ontologies. And if yes, what is the procedure that needs to be followed to request this addition directly to you or to the COPO team?
No, so as I said, the ontology bits of COPO are powered by a service called the Ontology Lookup service, which is hosted and developed by the EBI, the European Bioinformatics Institute in Cambridge in the UK. So I know that, so basically if you needed an ontology that's not already there, and I urge you to have a look at their website because they, like I say, they do have an awful lot of stuff already. OLS, oh, that's OSL. Oh, there we go. Right. So if we have a look here, we'll see all the ontologies. So here's all the ontologies they have, and they, you know, it's everything from life sciences to, you know, organic chemistry to astronomy to everything. So there's actually maybe I think they are all biology based, aren't they? They're sort of medical and biological.
We also have the crop ontology as well.
Yeah.
So, yeah.
So we had so our partners at the CG centres had their the crop ontology, different versions of the crop ontology added to this database. So you can now search for, you know, all stuff to do with crops. So if you have an ontology that isn't listed here, then you need to talk to Tony Burdett or Simon Jupp at the EBI about how to get your particular ontology.
Yeah. So I'll just press that link here so that if you wanted to take that further, you could know the people to contact there. So maybe from the home page here. So I think there is another question there which you could. Sorry, go on in.
Yes. So it's almost 12, and we should be ending now this webinar. I think that the best would be if there is any additional question from the participants, they could email you directly, since you could share your emails if you want in the chat box.
Yeah.
Or you could also
just to,
the questions directly to the IGAD website, so the IGAD mailing list, and you can capture these comments from there, right, Tony and Felix?
Yeah.
Yes.
So just quickly to answer Susie Robinson's question, sorry, we have a mechanism in COPO to add institutional repositories such as CCAN, DSpace, and Dataverse. So if you have a particular instance of those repositories that you need added, you can do so by getting the URL of the repository and your API key or username and password. So it's kind of agnostic as to which instance it submits to.
And quickly, Sophie, yes, you can restrict the ontologies, yeah, within COPO, yes.
Thank you so much for your time. This was really, really useful. And particularly in my case, I learned even more than in Finland because you had more time and we had the chance to really see more in detail how the tool works. So I really appreciate. Again, thank you so much for being here today. And to the audience, thank you so much for also being part of this webinar today. If you have any suggestion about any kind of additional topic that you would like to see in these IGAD webinars, please just email us and let us know. I wish you to all of you a nice day, and we keep in touch.
Great, thank
Thanks,
you.
guys.